at the front of the eye with the iris lying behind it, and the whites of the eyes, the conjunctiva, and the sclera surrounding it. And if you take tissue sections through this, okay, it's got the same bits as a human eye. Specialized skin cells in the corneal epithelium are dividing by immuno, and stem cells are label retaining because they are very slowly dividing. So that once they contain BRDU in their nuclei, they divide very slowly, and it dilutes out very slowly. Whereas a rapidly dividing cell, such as in the skin or over much of the ocular surface, will lose the BRDU really quickly because it's dividing all the time. It dilutes out the BRDU. So this slide just comes from an oldish paper that was labeling postnatal mice with BRDU, in this case for 4 days. And the BRDU label is shown in magenta. And you can see that after 4 days of BRDU labeling, nearly all of the cells in the ocular surface are labeled with BRDU. But then if you do a 4D BRDU label and then take away the BRDU, just give the mice normal drinking water for 9 weeks, most of the BRDU dilutes out, but you see a few label retaining cells identified by arrows here and there in the ocular surface. And what this paper showed, as I'll show on the next slide, if you map out where the label retaining cells are, this is a flat mount cornea, they're all around the edge of the cornea in the limbal ring. So this is how you identify that these stem cells are present in the limbus, because the only place where you find label retaining cells is the limbus. Okay, so that's old work, really, demonstrating how we identify the presence of stem cells in the limbus. And you may be asking, well, why am I telling you all this? What's the point? Okay, it's a good question. But the reason is it's relevant to aniridia-associated keratitis. What you're seeing on the screen here, this beautiful mouse eye that looks a bit like a sweet, is the result of an experiment that we did several years ago that was looking at patterns of cell maintenance in the mouse cornea. This is a female mouse who has an X-linked LAC-Z transgene. Now, LAC-Z, encoding beta-lactosidase, allows you to stain cells blue. And this female mouse has one X chromosome carrying the LAC-Z transgene. And because of random X inactivation during development, some of her cells are blue and some are white. And in most tissues, it's a horrible patchwork mess of blue and white cells. But in some tissues, it's not. And here you're seeing the corneal epithelium. And what you're seeing, these stripes of cells, is that in the limbus, around the edge of the cornea, there are patches of blue and white stem cells, and they are producing streams of blue and white corneal epithelial cells that are migrating into the cornea. Think about it. Every time you blink or get a bit of dust in your eye, cells fall off the corneal epithelium. Those cells have to be replaced, and substantially they're being replaced by stem cells around the edge that are producing streams of new cells that migrate into the cornea to replace those that have been lost. And this is actually essential for normal corneal maintenance. If the stem cells die, either by wounding or by, for example, alkali burns, industrial accidents, for example, then the cornea can no longer be maintained. You lose epithelial integrity, the cornea becomes opaque and effectively gets scarred, and people can effectively go blind. This horrible looking eye that you see on the screen here, I think, is a result of an alkali burn, I think. And the point is that aniridia-associated keratitis looks an awful lot like a limbal stem cell deficiency. Again, here is this is a 12-year-old kid with aniridia-associated keratitis, and the vascularization, the inflammation, the opacity, the corneal scarring and pannus looks like a limbal stem cell deficiency. And historically, and even today, aniridia-associated keratitis is treated clinically as a limbal stem cell deficiency. And the question is, one of the questions is that really true? Is that what it is? So we started a series of experiments to try and look at corneal maintenance in the PAX6 mice. And we did that by breeding PAX6 heterozygous mice with the blue mice carrying the X-linked LAC-Z transgene, so that we would get from these litters blue and white mice that were either heterozygous for PAX6 or homozygous wild type for PAX6, and we could compare them and see what we got. And the kinds of results we got are on this slide here. Here is a wild type eye with the blue and white striping that you expect, which, as I've said, reflects the migration of cells from the limbus into the cornea. And here are a variety, that these three panels are sort of representative of the sorts of patterns that you get when this LAC-Z transgene is on a PAX6 mutant background. And you can see it's really variable. You can get eyes on the right here that look more or less normal. You can get eyes on the left here that look very badly messed up. There is really no evidence of striping patterns. But most of them are somewhere in between, where you see patterns like this, where you can see evidence of a striping pattern, but it seems to have been badly messed up. So there is variability in corneal maintenance in the PAX6 mutant mice, which, as it happens, reflects the clinical variability in the disease. But clearly having PAX6 mutation doesn't preclude you from having reasonably normal levels of cell migration and presumably limbal stem cell activity in the cornea. Again, back in the old days, we made chimeric mice by aggregation of PAX6 mutant embryos and wild type embryos in vitro, and then reimplanting into pseudo-pregnant female mice in order to get chimeric mice, which are the things that you see here, with variable mixtures of wild type and PAX6 mutant cells, all very little nice cute mice. But we looked at their eyes, they were carrying LAC-Z transgenes as well, and we get this sort of pattern. Now, the difference between these eyes that you're seeing here and the eyes that were on the previous slide was that in this slide, the white and the blue cells can be different genotypes. So here's a control eye where with the white and the blue cells are both wild type, and you see normal patterns of corneal striping. The other three are eyes where the blue cells are PAX6 heterozygotes, but the white cells are wild type. And you're seeing relatively normal patterns of striping. So, for example, top right here, all these blue cells here are PAX6 mutant cells, and clearly the PAX6 mutants can form patches of mutant stem cells which migrate normally to the center of the cornea. So, given some wild type support, PAX6 mutant cells can behave perfectly normally in the cornea. And it's wrong to suggest that PAX6 mutant cells can't form limbal stem cells and can't migrate properly, because clearly they can. Some of the quantitative stuff we've done suggests that it may be that fewer clones of limbal stem cells are specified in PAX6 mutant mice, and that mutant stem cells are not as competitive as wild type in forming limbal stem cells. But PAX6 mutants do have stem cells, and these stem cells are capable of maintaining the cornea during homeostasis. So if there is a problem with these stem cells, what is it? And the other question that we addressed therefore was wound healing, because the dogma about these stem cells in the cornea, as in many other tissues, is that during normal tissue homeostasis, they aren't doing very much, that they divide relatively slowly, maybe only every 14 days, just enough to maintain the patterns into the cornea. However, when the cornea is a dose of another thymidine analog, chlorodeoxyuridine, CLDU, and then waited 24 hours, killed the mice, and then took their eyes for immunohistochemistry. And what she was looking for was double-labeled cells. Any label-retaining stem cells that had IDU, how many of them divided in injured and uninjured eyes in the 24 hours following a wound? And if the dogma was right, then what she would see is many more double-labeled cells in wounded eyes compared to unwounded eyes. The proliferation rate of limbal stem cells in injured eyes 24 hours after the injury. In uninjured eyes, only about 4% of